Hey guys, good morning. Happy Monday. I hope you all had a great weekend. Today and this week, we're going to continue on our investigation of protein synthesis and the mRNA molecule. We're looking at the different types of RNA and how each is involved in the synthesizing of a protein. So we are still in segment 5, so we're talking structure, function, and growth. And our essential question, our overarching essential question for a good part of this week is, how does DNA direct the making of a protein? So we're making proteins. So we know that proteins have various functions in the body and are essential to so many different things that are happening within our body. We know that genes code for traits, and those are proteins as well. And we're looking at our vocab. The third bullet point is protein synthesis. And just to note that synthesis, that means make. So making and synthesizing, those are synonymous. Those are mean the same thing. So when we say protein synthesis, we're really talking about making a protein. Our learning targets for today. So what you'll be able to do after this lesson and completion of this lab activity is we're going to use a model to explain how the genetic information in the DNA molecule is transcribed into mRNA. Learning target 2, explain how mRNA is translated into a specific sequence of amino acids in a protein. Lots of vocabulary within our learning targets. Transcribe, so we have transcription, translation. We've got mRNA, tRNA, amino acids. So while I'm on the topic of vocabulary, since you're all working on Chromebooks today for this interactive and have been for this phase of distance learning, it's kind of handy because you can go back and review your vocabulary organizers that you created electronically in your Google Drive. So if you go back, you can locate those in your Google Drive or go to Google Classroom. And if you need to, as vocabulary comes up and you're still kind of struggling with some of these terms, you can reference this where we made the Freyer models of the terminology for protein synthesis. So for example, and this is a student sample here. So for example, protein. So remember I had you guys go ahead and define it in your own words. You guys used the term in a sentence, and then I asked you to pull an example from biology specifically. And then we went a step further and we embedded a visual. So I had you guys look for a picture that would fit with the term and add that into this lower spot here on the bottom. And so we have mRNA, we have tRNA, and the visual is a really good option to put in because we can actually see the structure of the molecule and how it's different from the other molecules. We revisited DNA as well. So, and there's several more terms. So if you need to pull that up, I just wanted to remind you guys that you have this resource. So you created it for a reason. Use it, study from it, use it to complete this activity successfully. Alrighty. So went through the learning targets. So what are we going to do? So what's our agenda for the day? So I would like us to review some vocabulary. I would like us to look at an animation of the central dogma. So going from DNA to RNA to protein. We'll do a notice and wonder chart of this animation like we normally do. And what you'll be doing with the notice and wonder chart from this animation is contributing to a discussion post that I will post in a little bit later this afternoon. You'll have that option open and you'll see the instructions for that. You'll also have some processing time. So you'll be able to pause the screencast and process the notes from the last lesson. And this is really important, guys. So please process the notes on transcription and translation before you attempt to participate in the virtual lab. Okay, so processing those notes is going to be really important. And then the table and the journal questions are linked below the virtual lab under item 4A and B. So those are your documents. But again, remember you're going to Google Classroom to access those so that you can submit them. And then the bottom item students will need. So like always, make sure you have a pen or a pencil handy. You will need some notebook paper, your codon wheel, and the worksheets. The electronic worksheets are these documents here. So the codon wheel and the worksheets you guys have access to on Classroom. So your codon chart is what you will need to go ahead and complete the lab once you have your molecule of RNA synthesized. The post-lab journal question doc is here, and your link to the interactive. And remember your codon wheel. This is what the codon wheel looks like. So you can pull this up as you're nearing the end of your lab investigation so that you can reference this. Remember, you'll need to reference this to actually figure out which amino acid you will need to bring in for the corresponding code. All right, guys. So let's go ahead and look at some vocabulary really quickly. So the central dogma of biology. So I kind of just mentioned it. Reference your notes or just think back and see if you can recall on your own, how is this information passed? Just take a second and pause the video. Absolutely, it starts with DNA. So DNA we know is in every single nucleus of every single cell of our body. The DNA contains the genetic instructions for how we're built. DNA goes to RNA, that's the message. And therefore the RNA is translated into a functional protein. And when we talk about protein, the protein does something, okay? So it does something. Proteins have various functions that are essential to the body. So many different things proteins are responsible for. We're going to think of it as it shows up as a trait, okay? So we know that genes are segments of DNA, right, that are held within chromosomes. And the DNA is transcribed into RNA, and that RNA is translated into protein. And that's how you get the expression of the gene. So the gene that's held within the DNA is expressed as a trait by going through this process of protein synthesis. Okay, what about the process of going from DNA to DNA? So DNA replication or DNA synthesis, what's the term for that? Go ahead and pause and see if you can remember that and jot that down. What about going from DNA to RNA? So DNA is providing the template for an RNA molecule. And then what about going from RNA to making a protein? What's that process called? So absolutely. DNA making a copy of itself, that's replication. DNA going to RNA, that's what we call transcription, remember? Transcription. The DNA message is being transcribed into an mRNA, and that's our message. That's the message. That message, therefore, becomes translated into a protein. So really important vocab from last class. Hopefully you guys got all of those. Now is the time where you can go back and process those notes. So the notes from last class, guys, start here. So if you missed that last lecture on Classroom, it's not very long, but it's very important. So please look at Google Classroom, look for the lecture screencast from last Thursday, and take those notes, please. And if you have questions, any questions, please just email me or comment in Google Classroom. But you will need to have these done before we can go ahead and move on to our exploration today. So you've processed your notes. So I'd like you guys to do a notice and wonder chart. So if you could set this up on a separate sheet of paper, it doesn't have to be a lined paper, just a blank sheet, just a T-chart. Notice and wonder how we always do. And I'm going to show us an animation, a really cool animation in real time of protein synthesis actually, going from the molecule of DNA, so at the genetic level, okay, to a molecule of RNA and to a protein. What do you notice? What do you wonder? If you would like to go ahead and view this on your own, this is also linked to Google Classroom. You feel free to pause this at any time if you need to stop and collect your thoughts, to jot things down and go back, or rewind the screencast.
The DNA double helix contains two linear sequences of the letters A, C, G, and T, which carry coded instructions. Transcription of DNA begins with a bundle of factors assembling at the start of a gene to read off the information that will be needed to make a protein. The blue molecule is unzipping the double helix and copying one of the two strands. The yellow chain snaking out at the top is a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA letter by letter to copy the gene. At this point, the RNA needs to be edited before it can be translated into a protein. This editing process is called splicing, which involves removing the green non-coding regions called introns, leaving only the yellow protein-coding exons. Splicing begins with assembly of factors at the intron-exon borders.
Remember, you guys are completing your notice and wonder charts. So as you're looking at this, please be filling out what do you notice and what do you wonder,
which act as beacons to guide small proteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the two exons together. The edited RNA and intron are released and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes remove all introns so that the edited RNA contains only exons, which are the complete instructions for the protein. Again, this is happening in real time. When the RNA copy is complete, it snakes out into the outer part of the cell. Then all the components of a molecular factory called a ribosome lock together around the RNA. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. Inside the ribosome, the RNA is pulled through like a tape. There are different transfer molecules for each of the 20 amino acids shown as small red tips. The code for each amino acid is read off the RNA 3 letters at a time and matched to 3 corresponding letters on the transfer molecules. The amino acid is added to the growing protein chain, and after a few seconds, the protein starts to emerge from the ribosome. Ribosomes can make many proteins. It just depends what genetic message you feed into the RNA.
Okay, fabulous. So it just depends on the genetic message. So meaning it all depends on what is in the DNA. What is in the DNA? Okay, so to go ahead and, so again, wrap up your notice and wonder. Again, if you need to pause at any time and go back through this screencast, please feel free to. So the instructions for your virtual lab. So number 1, so go to Google Classroom and access the link, the table, the code on wheel, and your journal questions. So make sure you know where to go to find those items. Number 2, read the background information, objectives, and procedure for the lab. Number 3, follow the procedure to complete the steps of the lab. Number 4, complete the table. Number 5, complete the journal questions and submit. So I'm going to walk you guys through this. So if you go to Google Classroom, you have the link to your virtual lab right here. It will take you to a page like this. It will take you to a page that looks like this. So notice that I've also linked what you see here on the left, your background information, with your procedure and your objectives. So the background info is up here at the top, the first 3 or 4 paragraphs. Sorry, I don't mean to make anyone sick. Your objectives And then the procedure is really key. So I like that the procedure is on the same page as what you will be doing and manipulating with this interactive. However, I linked a Google Doc that makes each of you guys a copy. And this might be easier for some of you guys to read. This is background review information on the structure and function of DNA. We go into our objectives and then the procedure. So these are the steps of the procedure that you will be following to complete the task. So go ahead and pause this and please read your background info, your objectives, and your procedure, and then we'll be back together to get started. All right, so you read some background information. The objectives are restating our learning goals. So the one that's not highlighted is one that we've already discussed and tackled to describe the structure and function of DNA. You guys can do that and know very well the structure and function of the molecule of DNA. Now we're explaining how the genetic information within that DNA molecule is transcribed into mRNA, and going a step further, explaining how the mRNA is therefore translated into a specific sequence of amino acids. Specific sequence is key. Into a specific sequence of amino acids in the protein. So step 1, observe the unwoven DNA molecule. One of the DNA strands is exposed, showing a sequence of nitrogen bases. Again, our procedures right here, so you guys can read through these as you complete your steps. So we're looking at the DNA molecule, it says in step 1. It's all nicely structured as the double helix. And then we have our replication fork, and we see this part is unwound. And we see we have a list of nitrogen bases here. Those are DNA bases. And we have some corresponding RNA bases over here. How do we know they're RNA? Well, because there's a U. Remember that U is associated with RNA and it's not in DNA. So we need to match these bases over here. So what pairs with T? We know that A pairs with T. What pairs with A? We know that it's U in RNA. Reference your base pairing rules and synthesize that strand. Alrighty guys, so now you have most of your strands synthesized, so pay careful attention when you bring in your last base. You're going to be shown the simulation of what happens as protein synthesis is carried out. Pay careful attention, please.
Now, figure out the codes.
All right, so what we just saw was the mRNA molecule coming out of the nucleus. It found the ribosome, and the tRNA molecule carrying the anticodon matched with the codon on the mRNA, and it dropped off an amino acid each time. So if you hit show labels, this is going to help build your knowledge with the structures of this. Really important. So amino acids are represented by little circles, and each amino acid has a number, and each of you guys are going to be getting different sequences. So let me be really clear. The sequence that I translated here is not necessarily the sequence that you guys are going to be translating. This program gives everybody a different sequence. That's really cool, because essentially you're getting a different sequence of amino acids based on what the genetic code is telling is implementing in this process, is what I'm trying to say. So our mRNA molecule here. So this is your codon. So what's our next step? So what we need to do now is, and you guys can go through this several times. You might need to do it several times. You need to go ahead and go back to your rules. So you followed the procedure. Okay, and again, this is just a demonstration, so you guys can do this on your own. Complete your table. So you have your table down here. Alrighty, and so what I was presented with, my first amino acid is a number 1. So if you look at your table and if you look at your instructions, it says using the mRNA code deduced to the tRNA code. And if you go back up to your procedure, it gives you instructions to fill out that table. So you guys just follow your instructions. So I had amino acid number 1, and the mRNA code that it gave me for the first 3 amino acids was AUG. It was AUG. So I wrote AUG for mRNA. So now I need to figure out, well, what was the DNA code that initially started with? And then I need to go a step further and think, okay, so if the mRNA codon was AUG, what's the tRNA code? So you guys need to fill this part out. So I know that TAC corresponds to AUG because T pair is going to pair with A pairs with U, and C pairs with G. So now go ahead and follow your RNA base pairing rules to base pair and see what tRNA code you're going to get. I hope that's helpful. When you're done, click on your post-analysis lab questions and submit those. Email me with questions, and I will hear from you guys tomorrow. Bye-bye.